Welcome to Lesson 17.4, Molecular Evolution. In this lesson, we'll explain how new genes evolve, describe how Hox genes may be involved in evolutionary change, and explain how molecular clocks are used. One of the most basic questions we can ask is obvious. Where did the roughly 25,000 genes in the human genome come from? Modern genes probably descended from a much smaller number of genes in early life forms. So how could this happen? One way new genes can evolve is through duplication, followed by modification of existing genes. During meiosis, paired chromosomes swap DNA in a process called crossing over. Sometimes an unequal swap gives one chromosome in the pair an extra copy of one or more genes. If mutations occur in those duplicate copies, they don't affect the function of the original gene. This figure shows how this happens. The changing duplicate genes can gain new functions over time. Do genomes of living organisms contain duplicate genes? Yes. For example, the human genome contains 6 copies of the gene for beta globin, one of the polypeptides in the blood protein hemoglobin. Although these 6 beta globin genes are similar, mutations have slightly changed their characteristics since they were produced by duplication of a single ancestral globin gene. In other cases, gene duplication has allowed copies of genes to acquire completely new functions. This is how the antifreeze proteins of Antarctic fish arose. Chromosomes are not static DNA sequences. They can break and reconnect, lose bits and pieces of DNA, and acquire DNA sequences from viruses, microorganisms, and even other chromosomes. In fact, exons from one gene can mix and combine with exons from other genes. A sequence coding for an ATP binding region from one protein can combine with a sequence coding for a catalytic region from another gene, and a sequence coding for a regulatory region from still another gene. These changes can produce new genes that code for completely different proteins. Over time, gene duplications and rearrangements have greatly increased the size and complexity of eukaryotic genomes. They have also left evidence for evolution in the form of gene families. These gene families can be analyzed to trace the course of evolution and to help establish evolutionary relationships among different species. A cell does not usually create new proteins from scratch. What does this sentence mean in the context of what you've learned about gene duplications and rearrangements? Genomes use old genes for new functions. So how do gene duplications and rearrangements contribute to evolution? They contribute to the variation of alleles in a population upon which natural selection and genetic drift depend. Scientists think gene duplications are common, especially in eukaryotic genomes. The human genome, for example, is filled with duplicate genes. Some copies are simply relics. Others have been co-opted for other uses. Still others seem to interact with the original gene to produce enhanced function. Recently, scientists found evidence that an extra copy of a brain development gene in humans, which appears in our ancestors' genome about 2.4 million years ago, interacted with the original gene to allow maturing neurons in the brain to develop more connections. One exciting new research area is nicknamed EvoDevo because it studies the relationship between evolution and development. Darwin had a hunch that changes in the growth of embryos could transform adult body shape and size. Researchers have now shown how small changes in master control genes can produce exactly the kinds of evolutionary changes we see in the fossil record. As you learned previously, a group of regulatory genes, known as Hox genes, determines which part of an embryo develop into arms, legs, or wings. Groups of Hox genes also control the sizes and shapes of those structures. In fact, homologous Hox genes shape the bodies of animals as different as insects and humans, even though those animals last shared a common ancestor no fewer than 500 million years ago. Small changes in Hox gene activity during embryological development can produce large changes in adult animals. Insects and crustaceans are related to ancient common ancestors that possessed dozens of legs. Today's crustaceans, including shrimp and lobsters, have 5 pairs of legs, and insects just have 3 pairs. What happened to all those extra legs? Recent studies have shown that mutations in a single Hox gene, known as UBX, turns off the growth of legs in the abdominal region of insects. Thus, a small change in one Hox gene accounts for a major evolutionary difference between 2 important animal groups. The legs of the fruit fly and the legs of the brine shrimp in the illustration are the same color to highlight that a variant of the same Hox gene, UBX, directs the development of the legs of both animals. Each part of an embryo starts to grow at a certain time, grows for a specific time, and stops growing at a specific time. Small changes in the start and stop time can make a big difference in adult body shape. For example, small timing changes can make the difference between long, slender figures and short, stubby toes. Because changes in DNA help drive evolution, we can compare the DNA sequences of different organisms to determine their evolutionary relationships. If 2 species share a common ancestor, changes in their DNA should have accumulated since they become separate species. Biologists can use these differences as a kind of molecular clock to mark the passage of evolutionary time. Molecular clocks use mutation rates in DNA to estimate the time that 2 species have been evolving independently. To understand molecular clocks, think about old-fashioned pendulum clocks. They mark time with a swinging pendulum. A molecular clock also relies on a repeating process, mutation, to mark time. Simple mutations occur all the time, causing slight changes in DNA sequences. Some mutations have a major positive or negative effect on an organism's phenotype. These mutations are under powerful pressure from natural selection. Many small mutations, however, have no effect on phenotype. These neutral mutations tend to accumulate in the DNA of different species at about the same time. Researchers can compare such DNA sequences in 2 species. The comparison can reveal how many mutations have occurred independently in each group, as shown in the figure. The more differences there are between the DNA sequences of the 2 species, the more time has elapsed since the 2 species shared a common ancestor. So what evidence indicates that species C is more closely related to species B than to species A? Species B and C differ from each other by just 2 mutations, and they both differ from species A by 4 mutations. The use of molecular clocks is not simple because there's not just one molecular clock in a genome. There are many different clocks, each of which ticks at a different rate. This is because some DNA sequences accumulate mutations more quickly than others. Researchers choose a different molecular clock to compare great apes from the one to estimate when mammals and fishes shared a common ancestor. They check the accuracy of molecular clocks by trying to estimate how often mutations occur. In other words, they estimate how often the clock they have chosen ticks. To do this, they compare the numbers of mutations in a particular gene in species whose age have been determined by other methods.